my name is Vivi Tran, and today I'm here to talk about the cell cycle and how it is regulated. I will also be talking about how the loss of the control of the cell cycle can lead to cancer. So the cell cycle is a complex set of stages that is highly regulated. The cell cycle is divided into interphase, which is this part right here, and the mitotic phase, which is this yellow part. And the cell has to go through interphase before it can enter the mitotic phase. Since the cell spends most of its life in interphase is the longest phase of the cell cycle and it counts for 90% of it. You can see how in the diagram, interphase is the bigger chunk of the circle, which means that it's the longer phase. And interphase can be divided into three subphases. The G1 phase, which is this one, also known as first gap, the S phase, which is this one, also known as synthesis, and the G2 phase, this one, also known as second gap. And so during the G1 phase, a cell grows and carries out its normal functions, and then it enters the S phase where it copies its chromosomes. Afterwards, it enters the G2 phase where it can do some more growing before it can enter the mitotic phase, which is this. And the mitotic phase consists of two subphases, mitosis and cytokinesis. Mitosis is the division of the cell's nucleus, and cytokinesis is the division of the cytoplasm. The two genetically identical daughter cells that are produced from the parent cell at the end of cytokinesis can re-enter the cell cycle where it repeats. Mitosis. So the purpose of mitosis is to replace old cells with new cells. Mitosis consists of four phases. There is prophase, metaphase, anaphase, telophase, and cytokinesis is the division of the cytoplasm and follows mitosis. So during prophase, which is over here, the chromatin condenses into chromosomes, and the nuclear membrane and nucleolus break down. So the chromosomes are these X's. And then during metaphase, the chromosomes line up along the middle of the cell, which is right here. And then during anaphase, the chromosomes separate and move to opposite sides of the cell like this. And then during telophase, the cell forms two nuclear membranes around each set of chromosomes, and the chromosomes uncoil. So you can see that the X shape no longer remains, and then you see the nuclear membranes forms around each set of the chromosomes. And then during cytokinesis, which is here, a cleavage furrow is formed, which basically pinches a cell in two, so two new daughter cells can be created from the single parent cell. So you see how mitosis first started with one cell, and then at the end, now there are two cells. Ooh. Okay, so the cell cycle control system. So how does the cell know when to move on to the next phase? Well, the cell cycle is directed by internal controls or checkpoints, and these checkpoints have to make sure that the cell meets all of the necessary requirements before they can give the go signal for cell division. So you can think of these checkpoints like airport security, because airport security will only let you pass through the gate so that you can get onto your flight if you have met the requirement of not carrying anything dangerous with you. So if they notice that you are carrying a weapon or anything similar, they will not let you pass through the gate. And so the major cell cycle checkpoints include the G1 phase checkpoint, this one, the G2 phase checkpoint, which is this one, and the M phase checkpoint, which is this one. So the most important checkpoint is the G1 phase checkpoint, which is this one, because if the cell gets the go-ahead signal at this checkpoint and is allowed to go through the checkpoint, it can proceed to the mitotic phase where it can divide and produce two daughter cells. If the cell does not receive the go-ahead signal and cannot enter the mitotic phase, it will enter a non-dividing phase called the G0 phase. But in fact, most mature human cells remain in the G0 phase and never receive the go-ahead signal to allow them to go through the G1 phase checkpoint so they can divide. Such cells include muscle cells and nerve cells. And for the G2 checkpoint, which is over here, if the cell has damaged DNA, it will not receive the go-ahead signal at the G2 checkpoint. This will prevent the cell from entering mitosis to allow the cell to repair itself, and also prevents the creation of damaged cells. And then for the M checkpoint, which is over here, during mitosis, a cell will not receive a go-ahead signal at the M checkpoint when any of its chromosomes are not attached to spindle fibers. It will receive a go-ahead signal to allow it to proceed to anaphase when it meets the requirement of having all of its chromosomes attached to spindle fibers from both poles. So cyclins and cyclin-dependent kinases control the cell cycle. And kinases are the protein enzymes that control the cell cycle. They exist in the cell at all times but are active only when they are connected to cyclin proteins. So when the when the kinases are connected to cyclin proteins, they are called cyclin-dependent kinases, or CDKs for short. So how active CDK is depends on changes in the concentration with cyclin partner. So for example, you look at this graph, you can see that whenever the cyclin concentration peaks, and the cyclin concentration is represented by this darker purple straighter line. So whenever that line peaks, which is here, here, and here, the activity of a cyclin CDK complex known as MPF also peaks, and that's just an example for this particular picture. And so that the MPF activity is represented by this more curvy line, and so you see whenever this line peaks, this line also peaks. So in other words, when there is a lot of cyclin, MPF is super active. When there is not a lot of cyclin, MPF is not that active. So speaking of MPF, it is an example of how internal-external signals provide stop-and-go signs at each cell signal checkpoint. So MPF stands for maturation promoting factor, and it is a cyclin CDK complex. So we can also think of MPF as mitosis promoting factor because it controls the cell's passage through G2 checkpoint so the cell can enter the mitotic phase. So this diagram basically shows what MPF does. So cyclin is made towards the end of the S phase and continues through the G2 phase, and since cyclin is protected from degradation, it accumulates like this. And the cyclin then combines with the CDK, producing MPF, so then you see right here, it makes MPF. And when there is enough MPF, the cell passes through a G2 checkpoint, and that's a G2 checkpoint, and begins mitosis, which is this yellow M arrow. And MPF also helps mitosis by phosphorylating various proteins, which means that it adds phosphates to certain proteins. MPF is most active during metaphase. And during anaphase, MPF switches itself off by starting a process that leads to the destruction of the cyclin molecules, as you can see right here when the cyclin is degraded, and this ends the mitotic phase. And then the cell enters a G1 phase, and then during G1, the cyclin is further degraded like this, and then CDK, and then the CDK component of MPF is recycled like this. So PDGF. So PDGF is another example of how internal-external signals provide stop-and-go signs at each cell cycle checkpoint. So PDGF stands for platelet-derived growth factor, and it's made by blood cell fragments called platelets. So PDGF is required for the division of culture fibroblasts, which are a type of connective tissue cell, and when the PDGF binds to the PDGF receptors on the plasma membranes of the fibroblasts, a signal transduction pathway is triggered, and the cells are allowed to pass the G1 checkpoint and divide. And these cells rely on PDGF to give them the signal to pass that G1 checkpoint so they can divide and create more cells. So as you can see right here, in this culture, when there is PDGF, the cells are allowed to divide, but then when there is no PDGF, the cells are not dividing. So disruptions in cell cycle control. So why do we care about cell cycle control? Well, disruptions in cell cycle control can lead to cancer. So the normal cells that tell so the normal signals that tell normal cells whether to stop or go to this next stage of the cell cycle, that doesn't work on cancer cells. So you look at this diagram, you can see that the division of the normal cell, which is on the left, depends on the existence of the growth factors, you can see here. So if there are growth factors present, the normal cells will divide like this. But if there are no growth factors, then the normal cells will not divide like this. However, in cancer cells, which is over here on the right, it doesn't matter if these growth factors are present. So cancer cells will still divide uncontrollably in the presence or in the absence of growth factors. So there are some explanations to this phenomenon. The cancer cells may make the required growth factor themselves, or they may have an abnormality in the signaling pathway that conveys the growth factor signal to the cell cycle control system, even when the growth factor is not present. And a tumor is the mass of cancer cells that results. And a benign tumor is usually harmless while a malignant tumor is harmful because it can spread to other parts of the body. Well, that's it. Thanks for watching this video, and I hope this video helps. Bye!